Welcome to the Deep Dive. We take complex topics, dig into the research today, mainly drawing from Harper's Illustrated Biochemistry, and really pull out the core knowledge you need.
Exactly. We're not just skimming. The goal today is mastery.
And our focus. The incredibly intricate ways your body makes and uses fats. We're talking fatty acid synthesis.
Right. Our mission is to get beyond just the basics, to really grasp the details of how fatty acids are built and why they're so vital.
Things like energy storage, the actual structure of your cell membranes.
And even those potent signaling molecules that run so many processes, we're aiming for that deeper level of understanding, the kind that clarifies these really fundamental biochemical pathways.
And why should you care about how fat is made? Well, understanding this gives you a real window into energy management in the body, how cells are built, and even how chemical messengers control things like inflammation or blood clotting.
And there's some fascinating angles here, too, like how different animals, say cows versus humans, make fat from completely different starting materials.
Oh, right. And we'll touch on things like how aspirin works its magic at a biochemical level, which is quite surprising.
And also the critical difference between fats your body can make versus those you absolutely must get from your diet, the essential fatty acids.
So get ready. We're heading deep into the biochemical machinery of life. We promise to unpack the details, even the really complex bits, so you come away with a solid understanding.
Let's jump in. We'll start with the basics. De novo fatty acid synthesis, lipogenesis, making fat from scratch.
De novo, from new. Where does this primarily happen?
Okay, so the main location is the cytosol, that's the fluid inside your cells, and it happens in quite a few tissues: liver, kidney, brain, lung, mammary glands, and of course adipose tissue, your fat stores.
And the main thing being built is?
The primary product is palmitate. That's a 16-carbon saturated fatty acid. It's kind of the standard model built by this pathway.
Okay, 16 carbons and the starting material. You mentioned differences between animals.
Right. So in most mammals, including us, the main substrate is glucose, broken down to provide the carbon units. But in ruminants, think cows, sheep, they actually rely heavily on acetate.
Acetate, from where?
It comes from the fermentation happening in their rumen, their specialized digestive system. It really shows how metabolism adapts to diet,
Fascinating. So glucose for us?
Yeah.
Acetate for them? What else is needed for this synthesis to happen? It can't just be the carbon source.
Definitely not. It needs a whole toolkit of cofactors. We need NADPH, which provides the reducing power, the electrons for the building process.
Okay, NADPH.
We need ATP, the cell's energy money, manganese ions, Mn2 plus biotin, which is a B vitamin, and bicarbonate, HCO3, which actually serves as the source of CO2 within the reaction steps.
This isn't just theoretical biochemistry, is it? It connects to health.
Oh, absolutely. For instance, in type 1 diabetes, where insulin is deficient, this whole process, lipogenesis, is seriously impaired.
And on the flip side?
Well, how active this pathway is seems definitely linked to obesity. It's a central player in energy balance.
Okay, let's get into the nuts and bolts, the very first step you mentioned.
Right. The crucial first step, and really the control point, is making malonyl-CoA. This happens by carboxylating acetyl-CoA.
Carboxylating, adding a CO2 group.
Exactly. The enzyme doing this is acetyl-CoA carboxylase. And this enzyme, its activity level, pretty much dictates the overall rate of fatty acid synthesis. It's the gatekeeper.
The gatekeeper. And what does this gatekeeper enzyme need to work?
It needs energy, so ATP is required. And it needs biotin, that B vitamin we mentioned. Biotin acts as a carrier for the CO2 group.
And the enzyme itself? Simple protein?
Not at all. It's actually quite complex. A multi-enzyme protein. It has different parts. A biotin carboxylase part, a carboxyl transferase part, and something called a biotin carrier protein, or BCP, that basically swings the biotin back and forth between the other two active sites.
Wow, like a little molecular machine.
It really is. And it even has a separate regulatory site, an allosteric site, where other molecules can bind to turn its activity up or down.
Okay, so how does the reaction actually work, the mechanism?
It's a neat two-step process. First, the biotin carboxylase part uses ATP and bicarbonate to attach CO2 onto the biotin molecule. So biotin gets carboxylated.
Step 1, load CO2 onto biotin.
Exactly. Then step 2, the carboxyl transferase part takes that CO2-loaded biotin and transfers the carboxyl group onto acetyl-CoA, and boom, you have malonyl-CoA.
And this malonyl-CoA is the key building block for adding carbons later on.
Precisely. It's the activated 2-carbon donor unit for the rest of the synthesis pathway. And remember, because this acetyl-CoA carboxylase step is the first unique step and highly regulated, it's considered the initial and controlling step of the whole process.
Right, the bottleneck. Makes
Yeah.
sense. So we've made malonyl-CoA. What happens next? Where does it go?
Now it interacts with the next major player, the fatty acid synthase complex. This is another incredible piece of molecular machinery.
Another complex.
Yes, and it's a homodimer, meaning it's made of 2 identical large polypeptide chains working together. Imagine 2 identical multi-tool robots linked together.
And each of these identical chains does multiple jobs.
Oh yeah, each chain contains 6 different enzyme activities, plus a very important component called the acyl carrier protein, or ACP.
ACP, acyl carrier protein. What's its job? Sounds important.
It's absolutely vital. Think of it like a swinging arm or a tether. It holds onto the growing fatty acid chain and ferries it between the different enzyme active sites on the complex.
Like passing it down an assembly line.
Exactly. It ensures the intermediate is delivered to the right workstation for the next step. Functionally, it's similar to coenzyme A, which we see in fatty acid breakdown. And interestingly, the ACP itself contains a derivative of pantothenic acid, another B vitamin, in the form of 4-phosphopentethine. That's the actual tether part.
So the ACP is the delivery system within the factory, and you said the enzymes are arranged specifically.
Yes, the 6 enzyme domains plus the ACP are linked in a specific sequence along each polypeptide chain. And the whole dimer, when we look at its 3D structure through things like X-ray crystallography, it actually forms an X shape.
An X shape. Does having everything bundled together in this complex offer advantages?
Big advantages. First, it keeps all the intermediates close by, compartmentalized without needing actual membranes. It's highly efficient. All the substrates and enzymes are right there.
Okay, efficiency. What else?
Second, since all the enzyme activities on one chain come from a single gene, their synthesis is perfectly coordinated. The cell can turn the production of the entire synthase unit up or down with one genetic switch. Very streamlined.
Makes sense. A coordinated, efficient fat-building machine. Okay, let's walk through how this machine builds palmitate, that 16-carbon fat, step by step.
Right. It starts with priming the pump, so to speak. An acetyl-CoA molecule, our initial 2-carbon piece, gets loaded onto a specific cysteine residue sulfur atom on one part of the synthase complex, usually associated with the ACP of one monomer.
Okay, acetyl group loaded.
Then, a malonyl-CoA molecule, our 3-carbon donor, which will lose 1 carbon, binds to the phosphopentethene sulfur atom on the ACP of the other monomer unit. This loading is done by an enzyme activity called malonyl acetyl transacylase. Now the synthase is primed, holding both starting pieces.
Got it. Acetyl on one side, malonyl on the other. Ready for action. What's next?
Condensation. This is catalyzed by the 3-ketoacyl synthase enzyme domain. The acetyl group attacks the middle carbon of the malonyl group.
Attacks.
Chemically speaking, yes. And in this process, the carboxyl group that was originally added to make malonyl-CoA is released as carbon dioxide, CO2.
Ah, so that's where the CO2 from bicarbonate comes up.
Exactly. And this release of CO2 is energetically favorable. It really helps drive the reaction forward. The result is a 4-carbon molecule, a 3-ketoacyl group, specifically acetoacetyl, attached to the ACP, and the original cysteine site is now free.
So losing CO2 powers the joining step. Clever. Now we have a 4-carbon chain with a keto group. What happens to it?
Now comes a series of modification steps. First, reduction. That 3-keto group is reduced to an alcohol group, a hydroxyl group. This needs reducing power.
NADPH?
You got it. NADPH provides the electrons, and the enzyme is 3-ketoacyl reductase. So now we have a 3-hydroxyl group.
Ketone to alcohol using NADPH. Next.
Dehydration. A water molecule is removed from the 3-hydroxyacyl group. This is done by the dehydratase enzyme domain, and it creates a double bond between carbons 2 and 3. So now we have a 2,4, and 3 unsaturated acyl group.
Created a double bond, but palmitate is saturated, no double bonds.
Precisely. So the next step is another reduction. That double bond is reduced, saturated with hydrogens, again using NADPH as the electron donor. The enzyme here is anoyl reductase.
Second NADPH used. And the result?
The result is a saturated acyl group, but now it's 4 carbons long attached to the ACP. We've successfully added 2 carbons from the original acetyl-CoA and malonyl-CoA.
Okay, 1 cycle done, chain extended by 2 carbons. What happens to this 4-carbon chain now?
It gets transferred. This saturated acyl group moves from the ACP's phosphopantothene arm back over to that cysteine sulfur atom we started with. This frees up the ACP
And
arm
frees it up for what?
to grab another molecule of malonyl-CoA. And the whole cycle, condensation, reduction, dehydration, reduction, repeats.
Repeats how many times for palmitate?
Since we started with a 2-carbon acetyl group and end up with 16-carbon palmitate, we need to add 7 2-carbon units. So the cycle repeats 6 more times after the first one, each time adding 2 carbons derived from a malonyl-CoA.
6 more rounds. Wow, okay, so after 7 total cycles, we have a 16-carbon saturated chain attached. How does it get released from the synthase complex?
The final step is carried out by another enzyme domain within the complex called thioesterase, or sometimes deacylase. It specifically recognizes the 16-carbon palmitoyl chain and cleaves it off the ACP, releasing free palmitate into the cytosol.
And that's the end product. Is there an overall equation for this whole process?
Yep. If you sum it all up, it's Ul-1-acetyl-CoA plus 7 malonyl-CoA plus 14 NADPH plus 14H plus gives you 1 palmitate plus 7 CO2 plus 6 H2O plus 8 CoASH plus 14 NADP plus SALA.
14 NADPH is used for 1 palmitate. That's a lot of reducing power. And notice, 8 CoAs are released, 1 from the initial acetyl-CoA and 7 from the malonyl-CoAs.
Exactly. And it's worth remembering the first 2 carbons of palmitate come from that initial acetyl-CoA, the rest from malonyl-CoA.
So now we have free palmitate floating around. What happens to it? Does it just hang out?
No, it needs to be activated first, usually by attaching coenzyme A back onto it, forming palmitoyl-CoA. This activated form has several potential fates.
Like what?
It can be esterified onto glycerol 3-phosphate to make triacyl glycerols. That's our main form of stored fat. Or it can be elongated further, adding more carbons.
Elongated. We'll get to that. What else?
It can be desaturated, meaning double bonds can be introduced. Or it can be used to make cholesterol esters. Lots of possibilities depending on the cell's needs.
You mentioned a special thioesterase in the mammary gland earlier. What's different there?
Yes. The lactating mammary gland has a thioesterase that tends to release the fatty acid chain from the synthase complex earlier, when it's shorter, typically 8, 10, or 12 carbons long.
Why shorter chains?
These medium-chain fatty acids are important components of milk fat. They're more easily digested and absorbed by infants, providing a quick energy source. It's a neat adaptation.
Very clever. And what about odd-chain fatty acids? How are those made? We mostly talk about even-numbered ones like palmitate, C16.
Good question. Odd-chain fatty acids are synthesized using the same fatty acid synthase machinery. The only difference is the starting molecule. Instead of priming with acetyl-CoA, a 2-carbon unit, the process starts with propionyl-CoA, a 3-carbon unit.
So start with 3 carbons, then add 2 carbon units.
Exactly. Propionyl-CoA plus malonyl-CoA additions leads to fatty acids with an odd number of carbons, like C15 or C17. This is more common in ruminants, actually, because propion is another product of their rumen fermentation.
Okay, that covers the basic synthesis. Now, you emphasize the need for NADPH. Where does all that reducing power come from? 14 NADPH per palmitate is significant.
The major source, the workhorse provider of NADPH for fatty acid synthesis, is the pentose phosphate pathway. Specifically, the oxidative branch of that pathway.
Pentose phosphate pathway. I remember that runs parallel to glycolysis.
That's the one. And crucially, like fatty acid synthesis itself, the pentose phosphate pathway operates in the cytosol.
Ah, convenient. No need to transport the NADPH across membranes.
Precisely. And you see a high activity of the pentose phosphate pathway in the very tissues that are most active in lipogenesis: liver, adipose tissue, lactating mammary gland. The colocalization makes perfect sense biochemically. It provides the needed reductant right where it's needed.
Are there any other significant sources of NADPH?
Yes, there are a couple of others. One important one in many tissues is the conversion of malate to pyruvate, catalyzed by an enzyme called NADP malate dehydrogenase, or often just malic enzyme. This also happens in the cytosol and generates NADPH. And in ruminants, another enzyme, extrabiochondrial isocitrate dehydrogenase, seems to play a relatively larger role in supplying NADPH compared to other mammals.
So multiple routes contribute NADPH to the cytosolic pool for fatty acid synthesis. Now, let's track back to the other key ingredient, acetyl-CoA. It's the ultimate carbon source, right? Where does it come from mainly?
The primary source of acetyl-CoA is the oxidation of pyruvate, which itself mostly comes from the breakdown of glucose via glycolysis. And this conversion of pyruvate to acetyl-CoA happens inside the mitochondria, catalyzed by the pyruvate dehydrogenase complex.
Inside the mitochondria. But fatty acid synthesis is in the cytosol. Acetyl-CoA doesn't just cross the mitochondrial membrane, does it?
No, it doesn't. The inner mitochondrial membrane is impermeable to acetyl-CoA. So we need a transport system, an indirect way to get those acetyl units out into the cytosol.
How does the cell manage that?
It uses a clever mechanism called the citrate shuttle.
Citrate shuttle. I remember citrate from the Krebs cycle, the citric acid cycle, which is also in the mitochondria.
Exactly. That's the key.
Yeah.
Inside the mitochondria, an acetyl-CoA first reacts with oxaloacetate to form citrate. This is the first step of the citric acid cycle. But if energy levels are high and the citric acid cycle isn't running at full speed, this citrate can build up. And importantly, there's a specific transporter in the mitochondrial membrane, the tricarboxylate transporter, that can move citrate out into the cytosol.
Ah, so citrate carries the acetyl group, carbons out. What happens once citrate is in the cytosol?
Once in the cytosol, an enzyme called ATP citrate lyase takes over. It uses ATP and coenzyme A to cleave citrate back into acetyl-CoA and oxaloacetate.
There's our acetyl-CoA, now in the cytosol, ready for fatty acid synthesis.
Precisely. And this ATP citrate lyase enzyme, its activity is much higher when you're in a well-fed state. Lots of glucose coming in, lots of acetyl-CoA being made in the mitochondria. The cell is basically saying, okay, we have plenty of fuel, let's export some for storage.
Makes sense. But what about the oxaloacetate that's also produced in the cytosol by this reaction? It came from the mitochondria originally via citrate. Does it just build up?
No, it gets recycled back. First, cytosolic oxaloacetate is reduced to malate using NADH. Then this malate is acted upon by that malic enzyme we mentioned earlier.
The one that makes NADPH.
That's the one. Malic enzyme converts malate to pyruvate, and in the process, it generates NADPH right there in the cytosol, contributing even more reducing power for lipogenesis.
That's elegant. It helps transport acetyl-CoA and generates NADPH. What happens to the pyruvate formed?
The pyruvate can then be transported back into the mitochondrion. Inside, it can be converted back into acetyl-CoA by pyruvate dehydrogenase or carboxylated back to oxaloacetate to keep the citric acid cycle and the shuttle going. It completes the loop.
A beautiful integrated system. You mentioned the citrate transporter requires malate exchange.
Yes, the tricarboxylate transporter is an antiporter. To move citrate out, it needs to move something else in. Typically, it exchanges citrate out for malate in. This cytosolic malate going back into the mitochondria can then be oxidized back to oxaloacetate, ready to condense with another acetyl-CoA.
So it's a citrate out, malate in exchange. And going back to ruminants, why are ATP citrate lyase and malic enzyme less active in them?
It goes back to their primary fuel source being acetate. Since acetate is activated to acetyl-CoA directly in the cytosol, they don't rely nearly as much on shuttling citrate out of the mitochondria to get their acetyl-CoA. Less need for the shuttle means lower activity of the key enzymes involved, like ATP citrate lyase and malic enzyme.
Adaptation again. Fantastic. Okay, so we have built palmitate, C16. What if the body needs longer fatty acids, say C18, C20, or even longer?
That involves a separate process called fatty acid elongation. This primarily occurs in the endoplasmic reticulum, the ER. It uses a different set of enzymes, often called the microsomal elongation system.
Different location, ER versus cytosol, different enzymes. How does it work?
It takes existing fatty acetyl-CoAs, usually C10 or longer, both saturated and unsaturated, and adds 2 carbon units to them.
Using what is the 2-carbon donor?
Malonyl-CoA again, just like in de novo synthesis. And it also requires NADPH for the reduction steps involved. It's mechanistically similar to the fatty acid synthase reactions, but uses separate enzymes in the ER membrane.
So it extends the chain 2 carbons at a time, using malonyl-CoA and NADPH. Where is this particularly important?
A key example is in the brain, especially during myelination, the formation of the myelin sheath around nerve axons. There's a very rapid elongation of fatty acids like steryl-CoA, C18, to produce very long-chain fatty acids like C22 and C24.
Why such long chains in myelin?
These very long-chain fatty acids are crucial components of sphingolipids, which are major lipids in the myelin sheath, essential for its insulating properties and proper nerve function.
So we can build C16 from scratch, and then we can lengthen chains in the ER. Now, how is this whole process regulated? When does the body decide to make more fat or less fat?
The biggest factor is your overall nutritional state. Lipogenesis is fundamentally about converting surplus energy, especially from carbohydrates, into fat for storage.
So after a big carb-rich meal.
Exactly. When you're well-fed, plenty of glucose around, lipogenesis rates go up. It's part of the anabolic or building up phase.
And the opposite.
Conversely, if you're restricting calories, eating a high-fat diet, or if there's insulin deficiency like in type 1 diabetes, lipogenesis gets dialed way down.
Makes sense. Don't build fat if you're burning fat or can't process sugar properly. Is there a link to circulating fat levels?
Yes, there's generally an inverse relationship. When levels of free fatty acids in your blood are high, which happens during fasting, or on a high-fat diet, hepatic lipogenesis, fat synthesis in the liver, tends to be suppressed. It's a feedback mechanism.
If there's already plenty of fat available, don't make more. What about different types of sugar? Does sucrose affect this differently than glucose?
That's an interesting point. Sucrose, which is table sugar, is made of glucose and fructose. Fructose enters glycolysis downstream of a major control point, phosphofructokinase.
It bypasses a checkpoint.
Sort of. This means fructose carbons can flood the pathway towards pyruvate and acetyl-CoA more readily than glucose carbons can, potentially leading to a greater increase in lipogenesis compared to consuming glucose alone.
So the body is constantly sensing fuel availability and type to adjust fat synthesis. Is this regulation quick, or does it take time?
It's both. There's rapid short-term regulation acting on existing enzymes, and there's slower long-term regulation that changes the actual amount of the enzymes present.
Let's tackle the short-term stuff first. Modifying existing enzymes. Which ones?
The absolute key player for short-term regulation is acetyl-CoA carboxylase, that gatekeeper enzyme making malonyl-CoA.
Right, the rate-limiting step. How is it controlled quickly?
Two main ways: allosteric regulation and covalent modification.
Allosteric, meaning molecules binding somewhere other than the active site.
Exactly. Citrate, which signals abundant acetyl-CoA coming from the mitochondria, is a powerful allosteric activator. It makes the enzyme polymerize into long, active filaments.
So high citrate says go.
Pretty much. Conversely, the end products of the pathway, long-chain acetyl-CoAs like palmitoyl-CoA, are allosteric inhibitors. They bind to the enzyme and favor its less active dimeric form. It's classic feedback inhibition.
Product shuts down its own synthesis. Makes sense. What about the covalent modification?
This mainly involves phosphorylation, adding a phosphate group. Acetyl-CoA carboxylase is generally inactivated when phosphorylated and activated when dephosphorylated.
And what controls the phosphorylation?
Hormones play a big role here. The main kinase that phosphorylates and inactivates it is AMP-activated protein kinase, or AMPK.
AMPK. That's the cell's energy sensor, right? Activated when energy is low.
Exactly. AMPK gets switched on when AMP levels rise relative to ATP. It's also activated indirectly by hormones like glucagon and epinephrine, which signal low blood sugar or stress. They trigger a cascade involving CAMP that ultimately activates AMPK. High levels of acetyl-CoA can also help activate AMPK.
So low energy or hormonal signals for energy mobilization shut down fat synthesis by phosphorylating ACC. What about activation?
Insulin, the hormone signaling high blood sugar and nutrient abundance after a meal, promotes the dephosphorylation of acetyl-CoA carboxylase, likely by inhibiting AMPK or activating phosphatases. This flips the switch towards the active form, boosting lipogenesis.
So hormones and energy status fine-tune ACC activity moment to moment. Are other enzymes regulated short-term?
Yes, the supply of acetyl-CoA itself is regulated. Pyruvate dehydrogenase, the mitochondrial enzyme making acetyl-CoA from pyruvate, is inhibited by its own products, acetyl-CoA and NADH.
More feedback inhibition.
Right. Also, high levels of acetyl-CoA can indirectly inhibit pyruvate dehydrogenase by affecting the ATP-ADP ratio inside the mitochondria, and increased fatty acid oxidation, burning fat, also generates acetyl-CoA and NADH, which directly inhibit pyruvate dehydrogenase. It prevents the cell from trying to make fat while it's actively burning fat.
A coordinated shutdown. What's insulin's overall impact, summarizing its short-term effects?
Insulin really pushes lipogenesis forward. It increases glucose uptake into cells like adipocytes, providing more substrate. It activates pyruvate dehydrogenase in adipose tissue. And crucially, it inhibits lipolysis, the breakdown of stored fat, which lowers circulating free fatty acids and thus removes an inhibitory signal for lipogenesis.
Insulin is strongly pro-lipogenesis. Now, what about the long-term regulation? Changing enzyme amounts.
Right, this involves changes in gene expression. Key enzymes like acetyl-CoA carboxylase and fatty acid synthase are called adaptive enzymes because their amounts change based on long-term dietary patterns.
So what conditions increase their amounts?
Being consistently well-fed, especially with high carbohydrate intake, leads to increased synthesis of these enzymes. The cell gears up its fat-making capacity.
And decrease.
Prolonged fasting, high-fat diets, or untreated diabetes lead to decreased amounts of these enzymes.
What signals drive these long-term changes?
Insulin is again a major player, promoting the expression of the genes for ACC and fatty acid synthase. Glucagon generally opposes insulin's effects, suppressing their expression. And interestingly, certain dietary polyunsaturated fatty acids can also act at the gene level to suppress the expression of lipogenic enzymes.
So what you eat over days and weeks literally changes the levels of the machinery.
Exactly. These adaptive changes take time, hours to days, complementing the rapid short-term controls.
Okay, that covers regulation. Let's shift to a really critical topic. Essential fatty acids. We make palmitate, elongate it, but some fats we just can't make.
That's correct. Our cells have enzymes called desaturases that introduce double bonds, but they have limitations. We have D9, 6' and 4 desaturases, meaning they can put double bonds at those positions relative to the carboxyl end.
But not everywhere.
Critically, we lack the enzyme to introduce double bonds beyond carbon 9, specifically at the 12 and 15 positions. Plants can do this.
And because we can't make those, the fatty acids containing them are essential.
Precisely. The two main ones are linoleic acid, an 18-carbon omega-6 fatty acid with double bonds at day 9 and 12, and a linolenic acid, an 18-carbon omega-3 fatty acid with double bonds at 9, 12, and 15. We have to get these from our diet.
Linoleic omega-6 and alpha-linolenic omega-3, why are they essential besides just needing them structurally?
They are precursors for other important polyunsaturated fatty acids. For example, our bodies can convert linoleic acid into arachidonic acid, a 20-carbon omega-6 fatty acid, which is super important for signaling.
So if we eat linoleic acid, we can make arachidonic acid.
Mostly, yes. Though some animals, like cats, lack a key enzyme, the E6 desaturase, so they actually need preformed arachidonic acid in their diet.
Interesting. And fats like oleic acid, that's monounsaturated.
Oleic acid 18.1 Pa9 is non-essential because we do have an 8 to 9 desaturase. It can make oleic acid from stearic acid, C18 saturated. Similarly, it can make palmitoleic acid, C16.109 from palmitate.
So the 8-line desaturase is key for making our own monounsaturates. How does that enzyme work?
It's located in the endoplasmic reticulum membrane. It acts on saturated fatty acetyl-CoAs like palmitoyl-CoA or stearoyl-CoA, introducing that cis double bond at the 9-10 position.
What does it need?
It requires molecular oxygen, O2, and reducing equivalents, either NADH or NADPH. It works as part of an electron transport system involving cytochrome B5.
Okay, so we can make equinase, but for 6 of the 3s, we need the dietary precursors. How do we modify those?
Once we have linoleic or linolenic acid, our enzymes can elongate them, add carbons, usually 2 at a time in the ER, and desaturate them further, add more double bonds using those 6/4 desaturases.
And the new double bonds are added in a specific pattern.
Yes, they're always inserted such that they are methylene interrupted, meaning there's a single NECA-TISH2 group between the double bonds. That structure is characteristic.
So linoleic acid, too, can be converted to arachidonic acid.
Right. Linoleic acid 18.2 gets desaturated by A6 desaturase, then elongated, then desaturated by ethyl desaturase to yield arachidonic acid 20.4. Similarly, a linolenic acid 18.303 can be converted via similar steps into EPA, eicosapentaenoic acid 20.503, and then, though less efficiently in humans, further to DHA, docosahexaenoic acid 22.603.
So we process the essential precursors, but we can't make them from scratch. And is this processing regulated?
Oh, yes. The activity of these elongation and desaturation enzymes is significantly reduced during starvation or by hormones like glucagon and epinephrine or in insulin deficiency. It's tied into the overall energy state.
Why are these essential fatty acids, EFAs, so darn important? What do they do?
Their functions are diverse. Animal studies clearly show EFA deficiency causes poor growth, skin problems, reproductive issues. These are reversed by adding EFAs back to the diet.
Where do we get them typically?
Vegetable oils are often rich sources, especially linoleic acid. Alpha-linolenic acid is found in flaxseed, chia seeds, walnuts, canola oil. Fish oils are rich in the longer-chain omega-3s, EPA and DHA.
They have their functions.
2 major categories. First, they are essential structural components of cell membranes, especially in phospholipids, often at position 2. They influence membrane fluidity and function.
Structure. Okay. Second.
They are the precursors for a hugely important class of signaling molecules called eicosanoids. Think prostaglandins, thromboxanes, leukotrienes. Arachidonic acid and EPA are the main starting points for these.
Signaling molecules. We'll definitely need to unpack those. What about specific EFAs in membranes?
Arachidonic acid can make up 5–15% of phospholipid fatty acids, and DHA, that long-chain omega-3, is incredibly concentrated in the retina, brain cortex, tests, and sperm. It's vital for brain and eye development.
DHA sounds critical, especially for development.
Absolutely. It's supplied via the placenta and breast milk. Low DHA levels have even been linked to conditions like retinitis pigmentosa.
What happens biochemically if someone is deficient in EFAs? You mentioned skin issues, but at the membrane level.
The body tries to compensate by incorporating non-essential fatty acids from the thionine family, like mead acid, eicosanoic acid, 20.3 where the E phase should be. This changes membrane properties.
And can we measure this?
Yes, the ratio of this tonine train meat acid to the 2,6-tetraene arachidonic acid in plasma lipids. The train to train ratio is actually used as the biochemical indicator of EPA deficiency status. A high ratio suggests deficiency.
Clever diagnostic. Okay, let's dive into those signaling molecules,
Yeah.
the eicosanoids, derived from C20 fatty acids, you said.
Yes, primarily arachidonate 6 and EPA poo. Eicosanoids are like local hormones. They act near where they're made. The main classes are prostaglandins, PG, thromboxanes, TX, leukotrienes, LT, and lipoxanes, LX.
How do they signal?
They typically bind to specific G-protein-coupled receptors on cell surfaces, triggering downstream effects inside the cell. Very potent, even at low concentrations.
And the synthesis starts by getting the precursor fatty acid out of the membrane.
Exactly. Arachidonate, which is usually stored esterified at the second position of membrane phospholipids, needs to be released. This is done mainly by the enzyme phospholipase A2, PLA2.
PLA2 frees the arachidonate. Then what?
Once free, arachidonate faces a fork in the road. It can go down one of two major pathways, the cyclooxygenase COX pathway or the lipoxygenase LOX pathway.
COX versus LOX. What does the COX pathway produce?
The COX pathway leads to the prostaglandins and thromboxanes. Collectively, these are often called prostanoids.
And the LOX pathway.
That pathway produces the leukotrienes and lipoxanes.
Okay, let's follow the COX pathway first. Prostanoids. What's the key enzyme?
The key enzyme is cyclooxygenase, also called prostaglandin H synthase. It actually has two activities: a cyclooxygenase activity that adds oxygen and makes a ring structure, and a peroxidase activity.
Are there different versions of COX?
Yes, there are two main isoforms, COX1 and COX2. COX1 is generally considered housekeeping, present in most tissues all the time. COX2 is often inducible, meaning its levels increase significantly during inflammation.
And the product of COX?
The immediate product is an unstable intermediate called prostaglandin H, or PGH. PGH is then rapidly converted into the various specific prostanoids.
Like which ones?
Depending on the downstream enzymes present in a particular cell type, PGH can become prostaglandins like PGD2, PGE2, or PGF2, or prostacyclin, PGI2, or thromboxane A2, TXA2.
Different cells make different prostanoids.
Exactly. For example, platelets primarily make thromboxane A2, and endothelial cells lining blood vessels primarily make prostacyclin. This cell specificity allows for very localized control.
Let's talk functions. Thromboxane A2 from platelets.
TXA2 is strongly proclotting. It causes vasoconstriction, narrows blood vessels, and promotes platelet aggregation, helping to form a blood clot.
And aspirin's effect?
Low-dose aspirin irreversibly inhibits COX1 in platelets, thus blocking TXA2 synthesis. This is key to its anti-clotting effect.
And prostacyclin, PGI2, from endothelial cells.
PGI2 does the opposite. It's a potent inhibitor of platelet aggregation and a vasodilator, widens blood vessels.
So a balance between TXA2 and PGI2 is crucial for vascular health.
Absolutely. It's a constant push-pull regulating clotting and blood flow.
What about prostanoids made from the omega-3 precursor, EPA? Are they different?
Yes. EPA gives rise to the 3-series prostanoids like PG3, TX3. TXA3, for example, is a much weaker platelet aggregator than TXA2. PGI3 is still a potent anti-aggregator, similar to PGI2. The overall effect of shifting towards EPA-derived eicosanoids is often seen as less prothrombotic and less pro-inflammatory.
Interesting. So omega-3s might shift the balance. What other general roles do prostaglandins have?
Oh, many. They are major mediators of inflammation, causing redness, swelling, pain, fever. They're involved in modulating immune responses, regulating sleep, controlling smooth muscle contraction, like in the uterus during labor, protecting the stomach lining. The list is long.
But you stressed earlier, EFAs do more than just make these.
Critically important point. You cannot fix EPA deficiency just by giving prostaglandins. The membrane structural roles are separate. And inhibiting prostaglandin synthesis doesn't cause EPA deficiency.
Got it. You also called COX a suicide enzyme.
Yes, it's a way to describe that the enzyme tends to inactivate itself during the reaction. It helps limit the duration of the signal. Plus, prostaglandins are very rapidly broken down in the circulation by enzymes like 15-hydroxyprostaglandin dehydrogenase.
So their action is potent, but brief and local. Okay,
Yeah.
let's switch to the other pathway, lipogenesis, LOX. What does it produce?
The LOX pathway produces leukotrienes and lipoxygens. Lipoxygenases are enzymes that insert oxygen into a arachidinate at different positions, like 5, 12, or 15, to form hydroperoxides, HPPs.
And these lead to leukotrienes.
Specifically, the 5-lipoxygenes, 5-LOX enzyme, initiates the synthesis of leukotrienes. These are conjugated trienes often produced by immune cells like leukocytes and mast cells.
What do leukotrienes do?
They are powerful mediators of inflammation and allergic reactions. Some, like LTC4, LTD4, and LTE4, collectively known as the slow-reacting substance of anaphylaxis or SRSA, are potent bronchoconstrictors. They constrict airways, a key feature of asthma. Others, like LTB4, attract and activate immune cells. They increase vascular permeability, too.
So, very pro-inflammatory, involved in asthma and allergies. What about litoxins?
Lipoxygens are structurally different, conjugated tetrienes, and seem to have opposing effects. Evidence suggests they are actually anti-inflammatory and play a role in resolving inflammation. They act as counter-regulatory signals.
Fascinating. Pro-inflammatory leukotrienes and anti-inflammatory litoxins from the same basic pathway. Now, stepping back to the clinical picture, how do EFAs relate to chronic diseases?
Well, there's a lot of ongoing research, but epidemiological studies suggest diets higher in certain EFAs, especially omega-3s, EPA, DHA, are associated with reduced risk of cardiovascular disease, maybe some cancers, arthritis, and neurological issues.
What are the proposed reasons?
It could be through multiple mechanisms, altering the types of eicosanoids produced, for example, shifting towards less inflammatory ones, directly affecting cell membrane composition and function, or even influencing gene expression.
What about actual EFA deficiency in humans? Is it common?
It's rare in adults on typical diets, but it can occur in infants on very low-fat formulas or patients on long-term intravenous feeding without adequate lipids. Symptoms include skin problems, daily dermatitis, and impaired lipid transport, easily preventable with even 1–2% of calories from EFAs.
Are there diseases where EFA metabolism goes wrong?
Yes, abnormal EFA metabolism or levels have been seen in conditions like cystic fibrosis, ectodermatitis enteropathica, certain liver and kidney syndromes, Crohn's disease, alcoholism, and others. It suggests these pathways are quite sensitive.
And trans fats, where do they fit in clinically?
Trans fats, mainly from partially hydrogenated oils, are problematic. They can interfere with EFA metabolism. Structurally, they behave more like saturated fats and are linked to increased LDL cholesterol, decreased HDL cholesterol, and higher risk of heart disease. That's why intake should be minimized.
Okay, let's talk about drugs targeting these pathways. NSAIDs like aspirin.
Right. Aspirin, ibuprofen, naproxen, they all work primarily by inhibiting the COX enzymes, both EOX1 and COX2, thus reducing the production of prostaglandins and thromboxanes. This accounts for their anti-inflammatory, pain-relieving, and fever-reducing effects.
But inhibiting COX1 causes side effects.
Yes. COX1 has protective roles, like in the stomach lining. Inhibiting it can lead to gastrointestinal irritation and ulcers, a common NSAID side effect.
Which led to COX2 specific inhibitors.
Exactly. Drugs like celecoxib, Celebrex. The idea was to target TOX2, which is upregulated in inflammation, while sparing COX1 to reduce GI side effects. However, some COX2 inhibitors were later linked to increased cardiovascular risk and withdrawn or restricted.
And corticosteroids, like prednisone.
They work differently. They inhibit the expression of the COX2 gene, so less COX2 enzyme is made in the first place. They don't directly inhibit the enzyme activity like NSAIDs do.
So, various ways to dampen the prostaglandin system. Can we use prostaglandins themselves as drugs?
Yes, synthetic prostaglandins or their analogs have several uses. Inducing labor, treating peptic ulcers, managing glaucoma by lowering eye pressure, treating pulmonary hypertension. PGD2 is even being studied for sleep regulation. They have diverse effects, often by altering KMP levels in target cells.
And targeting the leukotriene pathway.
Also important, especially for asthma. Drugs that inhibit 5-lipoxygenase, like zilotarn, reduce leukotriene synthesis. Others, like montelukast singulaire and zafirlukast acylate, block the leukotriene receptors. They are key treatments for asthma and allergic rhinitis.
And maybe someday drugs based on the anti-inflammatory lipoxins.
That's an active area of research. Harnessing the inflammation-resolving properties of lipoxins is a very attractive therapeutic goal.
This has been incredibly thorough. We've gone from the basic building blocks like acetyl-CoA all the way to complex signaling molecules and clinical applications. Just to recap the big picture.
We saw how palmitate is built step by step by that amazing fatty acid synthase complex in the cytosol, requiring acetyl-CoA, malonyl-CoA, and lots of NADPH.
We saw how acetyl-CoA gets out of the mitochondria via the citrate shuttle, which cleverly also generates some NADPH.
We covered the intricate short-term and long-term regulation, centering on acetyl-CoA carboxylase and responding to nutritional status and hormones like insulin and glucagon.
Then the essential fatty acids, linoleic and alpha-linolenic, why we need them from the diet, and how our bodies process them into longer, more unsaturated fats like arachidonic acid, EPA, and DHA.
And finally, the eicosanoids, prostaglandins, thromboxanes, leukotrienes, lipoxans derived from those EFAs, acting as powerful local signals controlling everything from blood clotting and inflammation to airway constriction and immune responses.
Our goal was mastery, a deep dive into the biochemistry, and I think we've really covered the ground needed to understand these pathways at a high level.
Absolutely. The interconnectedness is striking. How energy status, membrane structure, and signaling are all linked through fatty acid metabolism. It's fundamental to how our bodies function.
So as a final thought for you, our listener, consider the fats in your own diet. How might the balance of saturated, monounsaturated, and the essential omega-6 and omega-3 polyunsaturated fats be influencing these intricate pathways within your own body? Or perhaps contemplate the future. How might further understanding of these eicosanoid signals lead to new therapies for inflammatory diseases or cardiovascular conditions?
The biochemistry is complex, but understanding it opens up so many insights into health and disease.
There's always more to learn in the fascinating world of biochemistry. Thanks for joining us on this deep dive.